Marine microbes are small organisms that make up more than 98% of the ocean's biomass. However, not much is known about the diversity of genomes within the microbes as depth increases. Researchers at the University of Hawaii at Manoa conducted an oceanic survey where they gathered water samples for over 2 years and separated the ocean depths down to 1,000 meters into 3 zones due to various characteristics. Overall, they researched the environmental drivers of a microbial genomic transition zone in the ocean's interior. This Biology 1511 project video is brought to you by Taylor Lee, Dylan Bloom, and Jimmy Fung. The overall purpose of this research were to determine the transition zones, observe environmental drivers of microbial communities, and correlate genome richness with ocean depths.
The research at the University of Hawaii found that there are 3 distinct transition zones that share similar trends but have various differences that isolate them from one another. The transition zones are classified by varying depths up to 1,000 meters that do not begin at surface level because of the large amounts of sunlight and the low amount of microbes compared to the other depths. The first zone was found to be from 75 to 125 meters deep. The second zone was from 125 to 200 meters deep. And the final zone was from 200 to 1,000 meters deep. The investigation found major differences in the environmental drivers of these 3 zones. Environmental drivers are any factor that may influence biodiversity in a particular ecosystem. The drivers were commonly were tested temperature, oxygen, carbon, phosphorus, nitrogen, genetic content, and genomic concentration. Genomic concentration is the amount of genetic material found in each zone by extracting and analyzing the water samples from the ocean in order to determine the quantity of microbial communities. The data suggests that nutrient variability, primarily nitrogen availability, across the 3 transition zones contributes significantly to species richness. Species richness, as we learned in Module 2, is the number of different species represented in a region. As we discussed before, this research found that there are 3 major transition zones. The transition zones are from 75 to 125 meters deep. The second zone is from 125 meters to 200 meters deep. The third zone is from 200 to 1,000 meters deep. The first transition zone had lower nitrogen levels and lower species richness. The middle zone had higher nitrogen availability and higher species richness. The lowest zone had low nitrogen levels and lower species richness. This means that nitrogen levels have a direct effect on the richness of the microbial communities. This figure illustrates the environmental drivers that are present in each of the oceanic transition zones. Part B of the figure shows the different factors and their relative amounts in each layer, while Part A shows a phylogenetic approach to sorting the environmental drivers. If you recall your knowledge on phylogenetic trees from Module 1, you will notice that there are 3 clades that all come from a common ancestor. Each of the 3 clades are shown from 75 to 125 meters, 125 to 200 meters, and from 200 meters to 1,000 meters. The figure shows predominant boundaries in the microbial communities between each transition zone, which causes a separation in the evolutionary history of these microbes. From this research, it was concluded that various environmental factors, especially nutrient availability, strongly influence the microbial communities at various ocean depths.